Hello,
Hello.
how are you?
Fine. I found a GitHub with Tensegrity on it. Um, Tensegrity programming, but it's programmed in Rust.
Okay, you have to learn
It
Rust.
isn't. It's another. Pardon?
You have to learn Rust.
Yeah, I guess.
Well, welcome, Susan and Shreyas and Kirill. Hello, glad you could make it. So, Kirill wants to present on his things. He's doing some C. elegans modeling, and that's
Okay.
so. Would you like to introduce yourself? So this is Susan's. So this is her link to the Tensegrity Lab software. This is in Rust.
Yes, Rust.
Okay.
Most of it.
Okay.
They said there was a bit of Python in there, but mostly Rust.
Yeah.
Yay.
Yeah.
It's about the 10th computer language I'm going to have to know.
Yeah.
This is ridiculous.
All right, well, we wait for Kirill, and Kirill's going to talk about his work on C. elegans modeling. So, if you're familiar with Asimov Press, they do a lot of popular science writing and things like that. And this is What's It Like to Be a Worm? And it's a pretty long read. I'm not going to go through the whole article, but basically it's about C. elegans, and it's about basically thinking about C. elegans behavior and what is it? Is it like just kind of mechanical behavior or a deterministic behavior? I mean, if you look at a lot of the research on C. elegans, one of the reasons people choose C. elegans to work on as a model organism is because it's very, you can trace it from a neural circuit to behavior that you can predict from the neural circuit activity and from the body movements. Everything's very stereotyped. And so that's an advantage of like looking at different types of behaviors. But then they have this question of what they call sentience, which of course is not limited to C. elegans. It's limited, it's not limited to organisms, biological organisms. It's limited. It's been applied more broadly to artificial intelligence these days. So this is just kind of goes through the history of C. elegans biology. And as it turns out, Darwin was really interested in some of the behaviors of C. elegans that were sort of, maybe sentient. And so he wrote a book on, or he wrote, I guess, a text on bioturbation, which is the reworking of soils by organisms. So, C. elegans moves around in nature, it moves around through a soil column. And collectively, they can basically change the composition of their home soil. So they change the composition of the soil, and they, basically eat vegetable molds. They eat like the bacteria in vegetables, rotten fruit, and other things. And then they basically excrete things and rework the soil by moving through the soil column. So this is something that was quite interesting to Darwin. And it's a reflection on animal sentience. This is the quote: "Judging by their eagerness for certain types of food, they must enjoy the pleasure of eating. Their sexual passion is strong enough to overcome for a time the dread of light. Notably, they're hermaphrodites, so for the most part. Although worms are so remarkably deficient in the several sense organs, this does not necessarily preclude intelligence. And we have seen that their attention is engaged. They neglect impressions to which they would otherwise have attended, and attention indicates the presence of a mind of some kind." That's a nice 19th century quote that, it's just kind of a reflection. There were no experiments done on that. Whereas Aristotle wrote about sentience in organisms like bees and in other types of organisms like fish. And what they mean by sentience, interestingly, is just basically those behaviors for which there's no immediate functional imperative. Is that really sentience, or is it something else? And this is probably the problem with the term sentience throughout history, is that you have this very ill-defined term, and it's maybe not quite, what we would like in a scientific context. And so this just kind of goes on thinking about some of the behaviors in humans that, is related to sentience. So this is an article on infant sense of pain. And then there's more stuff here on kind of linking sort of awareness to organisms like C. elegans. And so now we can explore the connectome, we can explore the neurophysiology of an entire brain. As small as C. elegans connectome is, it still can produce coherent behaviors, and perhaps that's a way we can sort of understand the neurophysiology of sentience, if such a thing exists. Then there's this term borderline sentience. While sentience is often defined as the capacity for pain and pleasure, this definition is incomplete. Sentience, in the sense we are using the term, so they kind of use the term, it's a philosophical version of the term. Basically, it's from this book, The Edge of Sentience by Jonathan Birch, and it's about the ability to feel pain and pleasure, and that's kind of where they kind of draw the line for this article. And then sentience is a combination of two things: consciousness and valence. To say an organism is conscious means that there's something it is like to be that organism, that is, experiences the world from its own subjective point of view. That is how philosopher Thomas Nagel puts it in his famous paper, What's It Like to Be a Bat?, which is a philosophical paper on this sort of, ascribing the features of batness to someone else, and being able to experience that. Valence is short for hedonic valence, from hedon, the Greek word for pleasure. To say a conscious experience is valence is to say that it's pleasant or unpleasant, that it feels good or feels bad. And so, we're looking for that type of thing. So this kind of just goes into the study of valenced experience. Ginsburg and Jablonka speculate that certain ancient animals, such as jellyfish, might experience a meaningless white noise sensation, an inner buzzing and crackling arising as a side effect of their decentralized nervous systems. And so this just kind of goes on about kind of reflecting on sentience and this more specific valenced behavior or valenced experience. And so and they get into things like persistent vegetative states and other types of autonomic functions, sleep-wake cycles, and thinking about, the boundary between awareness and non-awareness. So this is, and then they get into some brain organoid work, which is interesting. Neural organoids offer great potential for advancing neuroscientific research without the need for invasive experimentation on humans and other animals. At the same time, the pioneer of neuroethics expressed concern that in growing something like a portion of a human brain tissue, we might unwittingly be creating something like a portion of a human mind capable of positive and negative experience. Just kind of a point on just, talking about brain organoids and other types of systems as an alternative to like actual brains in a dish. And so then there's this limited behavioral evidence. Conscious experiences are private in the sense that they are not directly observable from the outside. When it comes to healthy adult humans, this does not pose too great a problem. If a research psychologist wants to know what a patient is experiencing, they can ask. It would be handy if we do the same with earthworms, but regrettably they can't speak. The same is true for comatose humans and neural organoids. And while we ask a chatbot or a parrot what it's experiencing, their answers are of little use as evidence, given that they have been trained to produce human-like responses without necessarily possessing the underlying subjective experience those responses typically indicate. This all goes to say that studying borderline sentience forces researchers to seek out and validate markers of sentience other than subjects' verbal reports. So a lot of sentience research has been reliant on self-reports, verbal reports, and we'd like to have a neurophysiological set of correlates for that. And so this just kind of goes through, how this might be done, and more parallels with humans, and then where neurophysiology can play a role here in some of the findings. So that's good. This is something that, run across in reading Asimov Press. It's a good article. There's a lot more detail, of course. So now Kirill is here. He's been in the OpenWorm Slack, asking questions, and especially in the DevoWorm Slack, and he wanted to go over his repository that he's been working on for simulating C. elegans. So I'm pleased to welcome Kirill, and he can introduce himself now, and then share his screen and talk about his project.
Hi guys. My name is Kirill. I work at NVIDIA, and I'm a software engineer. My specialization is graphics. So I work in a group called AI for Graphics. But in my spare time, I work on this project, which is a virtual cell, essentially. So I'm trying to create a model of living organisms in the computer. The C. elegans is a very good organism for that, because there's a lot of experimental data. So that's why currently I use C. elegans as the main test for my code. But it's not limited to C. elegans, so I do intend to go to other organisms as well. Basically what I'm trying to do is I'm just looking for some kind of experimental data, and then I try to simulate this experimental data in my repository. And right now, what I'm maybe a couple of months now, I was working on this C. elegans embryo demo. That so basically there is a video, and I'm sure you all guys saw this video, C. elegans egg, embryo, how it divides, how the cell divides. So I'm trying to reproduce this behavior in the computer. So this is the video, right? So this is egg of C. elegans, and there are two pronuclei. So there is a male pronucleus or female pronucleus, and they kind of move together.
Yeah.
Boom, and here they meet, right? So this happens at about 8 minutes time. So all this short sequence takes about 8 minutes. And here is my repository. And if I right now, this is what I have. So I have also two pronuclei. So I have an egg. Do you see it?
Yes, that's good.
So I have an egg. I have two pronuclei. And if I zoom in here, so this is a centrosome. So when sperm deposits this to the egg, it deposits the male pronucleus. This is male pronucleus, and there is a centrosome just near it. So they're here. Now, here in the left top corner, there is a timer. So this is simulation time. So if I start it, then what happens is that this centrosome, it starts nucleating microtubules. So those microtubules, they start growing, and what happens additionally is that in the cytosol, so in this liquid that's the cell filled in, there are dynein motors. And those dynein motors, they attach to those microtubules, and they start working on it. towards the centrosome, and this creates a pulling force on the centrosome. And this is the force that's working in this simulation, right? So this is the force that basically makes this centrosome moves around, right? And additionally, on the surface of this pronucleus, there are also dynamic motors. They also attach to the microtubule. So if microtubule comes close to the pronucleus, then there is a pulling force between the nucleus and the centrosome created by those dynamic motors. And this is what causes the nucleus to move as well. So and then at some point, the centrosome is duplicated. So there becomes two of it. So I can remove the microtubules. Now you can see that there is a second centrosome now. So let's do, and now they both kind of act on this nucleus. Then what's supposed to happen is that they are supposed to like migrate to different ends of the pronucleus because of the forces that act on them. So and now what happens is that those microtubules, since there are dynamic motors that are dissolved in this cytoplasm, the force on the long microtubules is greater than the force on the short microtubules. That's because simply there are more dynamic motors that can attach to the long microtubule. So this means that the centrosome starts migrating towards the in the direction in which it has longer microtubules. So those microtubules that are near the boundary of the embryo, they cannot grow too long, so the force is not as strong. But the force on those ends that are long, it's greater. That's why the egg starts. The nucleus starts moving in this direction. So now I guess it's hard to see what's going on because there are too many microtubules, so I'll just hide them. So now you can see just the two centrosomes and two nuclei. And now I'll resume the simulation. So now when those two nuclei, they are close by, what happens is that now those microtubules, they start also reaching towards this second nucleus. And there is also a force on the second nucleus, and that's why it starts moving towards the first nucleus. So now they move together. So and now they met, right? So this happens about 472 seconds. This is 472 divided by 60. So it's about 7.8 minutes. So it's about the same time scale that's on the video. So now after they meet, the simulation doesn't work correctly. So I'm working on the rest of the simulation. Yeah, so that's how it works.
All right.
And I have this repo in GitHub where so I sent invite to Bradley, and I asked maybe I can send invite to anyone who is interested, and you can guys you could go over it and give me some feedback and maybe tell me what is how it can be improved, right? So there's this is the repo. It's here, and I can, if you want, I can go over like general architecture of this and describe it.
Yeah, that would be great if you could do that.
Okay. Can you see can you still see the screen?
Yes.
Okay. So basically, there's a lot of code, and to organize this code, I needed some kind of hierarchy. So I created a very hierarchical representation of this, right? So the first level of organization is mostly by science. So everything that has to do with biology in this folder, then there's chemistry, there's geometry, physics, and that's it, right? And basically the organizational idea is that it has to be so that, for example, that it's like one folder can include the other folder, but not the other way around, right? There shouldn't be any circular dependencies. So this means basically, for example, the demos, right? The demos is just the executable. It's this screen that you saw right now that I was showing the worm embryo simulation, right? So these demos can include anything they want, right? So this is the top level. Then they include the viewer. The viewer is the everything responsible for rendering stuff on the screen. So the viewer cannot include the demos, but it can include everything else, right? Then the viewer, then the next level of this hierarchy is the biology. So biology, it's very like to simulate biology, we need all the sciences. So biology can include everything except viewer and demos, right? So it can include chemistry, geometry, physics, and so on, right? We can discuss. We can go into biology in more detail later, right? Then the next level is chemistry. So chemistry can include everything except biology and the viewer and demos, right? Then after chemistry, we have physics. That's the next level. So then geometry, and so geometry doesn't know about anything. It's just basically triangles and points, right? And then there's utils. Utils is just utilities that everybody use for their stuff. So this is the lowest level, utils. And then we can go into biology. So maybe this is because this is the biggest level, right? So biology is organized. So there is a top level, and this top level, it basically if like all other levels, except for example, the viewer accesses biology, typically it accesses this top level. So this is the level that everybody sees, right? And there is a cell simulator and organism. And the important class, it's called data collector. So this data collector, it's used for debugging. It's what it does basically. It can you can say I wanna like every simulation step I wanna collect this and this data, like the concentration of some particular protein, for example, right? And then you get as an output a CSV file that contains this data, right? And I think every time I run the simulation, it's like so these folders here in the data sim output. This is the this folder is created every time I run the simulation. So right now I was running, this folder was created. And there is a CSV file that's created, so you can see it saved some data. So there is a time, there is this is time step, this is simulation time, and some data here. So you see there is, I was looking at gamma tubulin concentration. So there is gamma tubulin RNA and gamma tubulin protein. And I was looking at it because it affects the number of microtubules that are created on the centrosome. And this is a centrosome chromosome. Oh, I'm not sure what's the chromosome condensation. So one important thing maybe to note is that most of the code is created using AI. So basically, it's a vibe-coded repository. So I don't know a lot of the stuff that's here because I rely on AI to know a lot of biology instead of me. So like this chromosome condensation, for example, I'm not sure what it is. I asked the AI that I need to debug something, and it just added this, so this column. That's why I don't know what it is. All right, so I was describing this top level. So this data collector is the class that saves this comma-separated file on every simulation. And basically what happens is that when I debug some particular thing, I'm telling to AI, please modify data collectors. We need to collect this and this data. So it modifies it, and then I run simulation. I get a new CSV file I can look at. So then the logic here is that we have biostate. So this biostate project, it basically contains the state of all biology. So the idea is that I separate the state and the processes that occur in the cell. So mostly I'm trying to have here in this folder biostate, I try to have a very small amount of functions. So these are just this is just data, right? Just data that represents the state of the cell, and all the functions are here in the folder that's called processes. So there is like if we open it, we can see, for example, there is a centrosome process, envelope state process, microtubule dynamics process, and so on, right? So these are all processes. They just run every simulation step. They run one after another, and they modify this biostate in some way. And the last project that's here is called Forces. So this is basically sorry, this is wrong. This one. So these are all the forces that affect the simulation, and the reason it's there is because I have physics, right? And normally forces should be in the physics. These are basically adapters. So this project physics, it cannot know anything about biology, so it operates with stuff like orientational spring, for example, or a mesh, triangle mesh. So it doesn't know anything about biology. So for example, the force. If we look at the force, it has just a virtual function here. It says apply. So the force has to apply itself. And then this function would be implemented here. For example, if we look at this, so implementation of one of the biological forces, then it implements this function apply, right? So it says, okay, this is a microtubule cortex force, right? So and it applies it. And then the physics engine, it doesn't know that this force is related to microtubules. It just applies it to a sphere, which is a nucleus, right? And again, it doesn't know anything about nucleus. It just sees a sphere, right? So this is just data in the function. It's just organizational thing. So I guess that's the level I wanted to go into. So if you guys have some feedback right now or want to share it later, then go ahead.
That's great. Thank you, Kirill, for that overview. I know we have questions, so yeah, I think it's great. And we talked a lot about how to think about these processes in the meeting. So like one of the things with cell division, how deeply do you want to model it? Is it good enough just to have a model of cell division where you're just counting cell divisions and have like this blob that divides? or do you need to have a lot of biochemistry simulated underneath, or what's the specificity? So I like that you have the specificity there. We've done things with like CompuCell 3D, which has kind of a different approach to simulating cells and cell division. But it's like you always have details that you want to investigate. So it's always good to have different models that can simulate those things from data, because that's we usually have like secondary data sets that we mine and then kind of put them into simulations or analyze them in some way. So that really does give you a lot of flexibility if you can bring in data sets and benchmark things like that.
So you just mentioned some package that you were using. What I didn't catch it. What was it?
So yeah, we've used a number of packages. One that of interest to at least some of us is called CompuCell 3D. That's a package. I think it's written in C, but there's a community around that where they use it for very general purpose biology. But of course, you can use it for development, but it's like not specialized for development. So you can, it's James Glazer is the head person on that.
Oh
project.
I took a couple courses under him during my undergrad at IU. Him as well as I did my undergrad at Indiana University Bloomington. So there's him, James Glazer. He was there from CompuCell 3D. There's also Dr. Robert DeRuyter from the physics department. And he would do pivotal research on spikes, which is basically the book that was one of the seminal books of computational neuroscience from physics. Yeah.
And what kind of things could you simulate using this package?
Using CompuCell. So with CompuCell 3D, we did some pretty basic agents with different types of cellular structures, single cellular, multicellular, seeing how they themselves would respond to differences in environments, mostly dynamics, mostly biophysical dynamics, given constraints on the sorts of decision making matrices, minimizing or sometimes maximizing like resources, energy available to cells and available to the agents in the environment.
Also, cell sorting.
Cell sorting is important.
You can do cell sorting with it. It's an agent-based model. It's kind of.
What is a cell sorting? I'm sorry, I'm not familiar with this term. What does it mean?
Sorting according to surface tension. And you can get cells of the same type to clump together, and some cells will surround the other cells. So there's a sort of so you can sort of make an organelle using it. You can start with a bunch of different cells, and they'll sort together or sort in kind of like an organelle.
Is it still in the context of C. elegans, or like?
It's a physics based and chemistry based modeling.
But it's still modeling of the organism C. elegans or different organisms.
Just cells.
Yeah, it's a generalized program. So like a lot of the simulation platforms, most of them are, you can model generalized processes. Of course, we also want to have like specialized models because we have very specific modes of development in different organisms. Like C. elegans has the lineage tree that you're building from. So we want to be able to capture that. Whereas in other organisms, the lineage tree is different, or there's a different kind of way that's done. And so having like capability models and also kind of a general capability is really kind of nice in a model. You're capturing maybe some very specific things in, say, C. elegans or Drosophila, and then you also have like generalized processes that you can simulate. So you can switch them between models. Like the biophysics is generally similar. So, that's something that goes across development, but then you have very specific things in a species, in a model organism that you want to get at.
Yeah, it didn't help me with my project, which was tensegrity of epithelial cells.
Wow.
So it wasn't 3D enough for me, I guess you'd say.
Yeah, and then there again, like with that's a problem of simulations in general, that you can't get, like, it's not a typical biologic, or it's not a typical physical model. But yeah, I mean, I don't know how much you've interacted with OpenWorm people, like seeing what they have in terms of models.
Well, I met with Steven Larson. He was just showing me so they have this website where you can look at this 3D simulation of a worm, and you can look at different structures inside the worm. And I also he showed me some package that where a worm would be simulated, so it would be like wiggling in some kind of volume filled with spheres. It's called cybernetic, I think.
Right.
And yeah, so I will. So my goal was to achieve something that we basically from the full development of a worm, starting from this one embryo, so that it develops, then it has to split to 2 cells, then more cells, and so on, and then in the end the worm would come out, right?
Right.
And it has to be like. The simulations should be good enough so that it would allow some experimenting. Like if you did some experiment on the virtual worm, and then you did the same experiment on the real worm, you would see similar results. So it has to have some predictive power, right?
Yeah.
So do you know how far, because you have different models, right?
Right.
Different kinds of models that you are working on. So what kind of predictive power do your models have?
See, for C. elegans in development, we're fortunate because C. elegans has a lineage tree that is deterministic, meaning that we can say, we can build a model where we divide, and we have cell divisions, which are doublings of a cell, and then we know kind of where those cells are in the embryo and how those divisions should proceed. So you start with one, the one cell goes to 2 cells, and then you get to the founder cells, of which you have 8, and then they start to differentiate into germline, and that's where you get asymmetric divisions. So we've built models around looking at the sort of the connectedness of cells, the sort of categorization of cells, and that whole process of building a phenotype from the cells, meaning that the cells are dividing and differentiating, and they're forming sort of structure. And so that's kind of the way what we focused on. We've not had something of this detail where you're looking at specific events in cell division and then kind of building up to that. I like that idea of having focusing on the biophysics, and then it's you can expand that out to the entire process of development. But we don't have anything of that scale. We just kind of focus on the sort of the process of building a phenotype. So you have cell divisions from data, and it's at the level of the cell, and then where those locations are in the embryo, and then how those build to a phenotype. So
I'd say a couple of things. There are many, many, many types of models out there. It's kind of the Wild West, and that's quoting James Glasner, saying it's the Wild West. Yeah. So if you're interested in CompuCell 3D, it's on YouTube, and he gives a course in it every summer. So you can go attend it. I attended it a couple of years ago.
Yeah.
No, well, I
It was
definitely will look it up.
Yeah.
Okay.
So I was wondering if it's possible to write like one best model that like simulates everything.
Well, that's my philosophy is probably not. I mean, there different hybrid models that I would probably advocate for. Meaning, like you'd have like models that do different things in development, and then kind of making those work together. So it's like well, with OpenWorm, one of the things about what they're doing in the main project is they have all these parts, and they're simulating like the connectome and the biophysics, the cybernetic model, and then they have a viewer, and those are all kind of, you can have those, and you can get a certain understanding of the worm. You can run experiments, but they're still kind of disconnected. In other words, you can't build like it's really hard. It's harder to build an integrated model of the worm. I mean, it's important to focus on the different things going on, like the physics and the neurophysiology and things like that. But then integrating all those is very hard. Not only because it's like you have kind of different imperatives with each type of model, but because it's like you have to really kind of have the same framework, exactly the same framework to get things to work. You were showing how you have different modules in your model, like where you have you're running some process, and for biology or for physics, and you have a physics engine, and you're doing that, and then you have the biology, and you have that. You have to coordinate all of that across the worm. So I don't really think that. I mean, I think that's a very long-term goal to do, like sort of a single model that integrates everything. To have different models giving different insights, that's maybe better. But I think it's also, I like hybrid models where you can try to get them to work together in some way, or you have some heterogeneity in the modeling, but you're kind of working towards a common goal. It's I don't know. We've tended in this group to.
Why do we need a single model? Hey Bradley, why do we need a single model?
Well, there is this vision that people have of simulating the worm, and so like that's where people have said OpenWorm has succeeded or failed, even though that's not really, the people in the project don't really think that. But I mean, I guess the idea of a single model is like people think of development as this single thing, right? Development of C. elegans is a single thing that happens, and we observe it, and underneath that is all this complexity. And the single model is just kind of like replicating that. But replicating the parts, of course, is important to understanding it. They don't need to be a single. They can be all sorts of different models. But
I mean, I mean, fundamentally, it's not. I'm not actually so concerned with, say, accounting for all sorts of types of information, like the chemistry, the biology, the physics, the genetics. But more so with whatever models you're using, like OpenWorm being like a digital twin. You guys know what a digital twin is, right? When you so like an OpenWorm being like the digital twin, but also like some of the more like biomechanical ones, like CompuCell 3D, very much like biophysics biomechanically. Yeah. What I was what I was to what extent can the rules be applied from one to another and modeled in the same way? So like if X then Y, right? So if something, then like if you're exposed to this condition, this environment, and the cells would divide following these rules or something like that, right? And from a single model perspective, or generalizing from one to another, I guess I guess there's this question: to what extent are the models that we are building, like OpenWorm and CompuCell 3D, to what extent are they themselves meant to describe things as they are, or are they more so meant to be, this is what we know, and we in some ways are trying to see how what we know would make sense in some sort of other environment, right? So are the models themselves, do they all have some sort of truth to them that we're trying to combine and generalize and make and see how they relate to one another, or do they all represent some sort of similar idea, similar picture, similar goal, similar ground truth that we are all describing in a different manner, you know?
Yeah.
And that's a really interesting question to think about. Like that's what I think about with these models. So yeah.
Well, I think the thing here is that, like, when we build a model, we usually build it from what we know about biology. I mean, that's what we have to do because we don't, and we put data into it. But sometimes in biology, we discover new things, and it upends what we know about some process. And sometimes that's like just kind of details and doesn't necessarily matter. And sometimes it's the big picture. Like, I don't think our knowledge of C. elegans development is going to be upended tomorrow. But there's, like, we've been talking in the group about sentitium, sentitial cells. And so that's something we don't necessarily we can do like a simulation of that. That's where cells.
What is sentitial cell?
So this is where you have cells that are born and then they merge or they fuse together. And so, like with muscle, not in C. elegans, but in muscle, you get cells that fuse together. And so you get a bunch of nuclei in a cell body. And that's
so it becomes one cell?
Yeah.
Oh.
And and then there's this there are these dual processes of like nuclear dynamics where nuclei are dividing and the cell is doing, some sort of function. And muscle, it's trying to, establish twitch dynamics and things like that. But so there are reasons why sentitial cells kind of exist, but we don't exactly know why and how they form the way they do. So we could simulate that, but we might be wrong in terms of how they form, and we might get the parts right. We can simulate the nucleus, we can simulate the cell fusion, and then maybe some of the functions there, but like we might not be have a very good handle on how maybe why this is happening or all the varieties of ways in which that can happen and things like that. So I think it's. Go ahead.
So my opinion is that we need to go and simulate on the lowest possible level that we can, right?
Yeah.
For example, if I try to simulate right now on a level of atoms, it will not be practical, because there are too many of them, right? So that's why I'm not doing that. But if I could, of course, I would go lower. It's just I don't have enough compute to simulate that.
Right.
And the reason we need to simulate as low as possible is because we want to get emergent behavior. Like if I just say, if I have the hair, for example, and I move this hair over the worm, and it needs to twitch in some certain way, and if I just program this behavior, right, I just say, if the worm is touched in this place, it has to twitch. Then it's not emergent. I programmed this behavior, so of course it behaves this way, right? So it's not very interesting for understanding and for to getting new knowledge. So the point of this exercise is to get some new knowledge. So you simulate on a very low level. From this you get some emergent behavior, and you compare this emergent behavior to the real world. Like if the real world also behaves this way, this means you've got some detail of simulation right? And this is what guides the simulation. You simulate, you compare. If it's not right, you try to figure out what you did wrong in the simulation. That way, over time, the model becomes better and better and better, and it becomes really you get a lot of new knowledge out of it. Right.
Yeah.
So that's why, and that's why I think there could be one model, because, if the goal is to go the lowest possible level, then it's enough essentially to have. It's just one goal, right? So the goal is to have this lowest level. So there could be one model that goes lower than any other model, and then it could be the only model you need.
So if there is just one model, or if you're going to make one, it needs to include tensegrity. There we go.
What is tensegrity?
It's a structure that's made out of rods and strings, It's held up by tensioning the strings. And the reason why tensegrity is a part of biology is because all of our cells are in tension, and they operate like a tensegrity on the cellular level and also on multiple scales of levels in between, including bone and muscle, could be considered a tensegrity object.
Yeah, for sure. Yeah, it sounds like I need tensegrity. Yeah.
Yeah, I did my master's thesis on tensegrity of epithelial cells.
So, like, for example, the worm, it would have tensegrity, right? Because, like, when all these muscles, yeah,
the muscle and the skin and all its organs. Yeah, yes, basically.
Yeah.
Tensegrity is a principle. Yeah, I guess.
Well, I think what Kirill is like. I think what's interesting about that
tensegrity being a structural principle. So I guess anything with a structure would have its own tensegrity, whether it's a building, whether it's a worm. So yeah.
But I think what was interesting about what Kirill just said was that, you think about like DNA as being the underpinning of all biology, or proteins as being the underpinning of all biology. If you had a model of DNA or proteins, you could build theoretically like all these different systems and generate like the structure of an organism. Now, we don't necessarily know how that, like, all those processes are very heterogeneous, but you have that lower level model that's, at least in part predict all of those other things. So, yeah, I think in that sense,
I'm sorry, DNA is a tensegrity. Microtubules are a tensegrity. Anyway,
okay, but then I already have tensegrity because I have microtubule.
Yes, yeah, exactly.
Just to be clear, by the way, not everything has tensegrity. Like it needs so if something like say expands or changes in form in a way that's I guess okay, like something that's like continuous, right? Something that's continuous and doesn't rely on that sort of structure on how it operates. So like I guess like tensegrity itself has to be isolated, like bones, like the structures of like a bones and a skeleton, yeah, and they have to be.
It needs it needs the tensioned elements and the compression elements.
Yeah.
And the tensioned elements aren't necessarily elastic. Like they're more like a string than an elastic to operate properly. So it's they're otic. They don't have a proper Poisson's ratio if you're getting into mechanics. They're otic.
Okay.
All right. I actually.
So returning back to this one model, right, discussion. So I was thinking, so let's say we want to build one model that goes as low as possible, right? Mm-hmm. And it's probably very large amount of work, right? But I think so. Maybe it's not achievable, but I think it's a interesting goal. And like if we look at this repository I was just showing you, right? We can look at it as some kind of organism that develops over time, like in like biological organisms, they experience evolution, right?
Yeah.
So and we kind of think of ourselves as the force of evolution. So we go and do this repository. We say, okay, we do very small change because evolution goes in very small steps, right? So we do those small changes that make it better and better and closer to biological reality. That way, this organism kind of evolves, and over time, if we have enough time and enough persistence, it will just achieve something, right? It will become great.
Yeah.
It will be able to simulate everything.
Yeah.
And also simultaneously with this, because it needs a lot of compute, right? So essentially, if you want to simulate something on low level and something big, then you need a lot of compute power. But right now it's a great time because everybody is building compute for AI, right? So there are trillions of dollars invested into expanding of compute and building data centers, right?
Yeah.
So and compute becomes cheaper and cheaper and cheaper. So simultaneously with model becoming bigger, the compute will become bigger. So it kind of will all scale together.
Yeah.
Here's a tensegrity. It demonstrates otic behavior. If I push on it here, the whole thing contracts. And if I pull on it, the whole thing expands. And if you have other types of material, you'll have Poisson's ratio, where if it thins in one direction, it expands in another. This does not. It compresses all together when you push on two sides. And when you pull on the sides, the whole object expands. Like it works, it's connected all together. And it moves all together.
Okay.
Yeah, that's my best explanation right at the moment.
I will interrogate AI about how it can help me in this development. Maybe, yeah.
Looks like Morgan maybe wanted to say something. Did you want to say something, Morgan? Or
sorry, I just got back from dropping my daughter at school.
Okay.
But I was my one comment I put in the chat just about, to Hussein's point about, what's the kind of scope of the models that you're interested in. I like describing them like organ on a chip experiments, where it's just like you're not actually making an organ, you're actually, kind of fabricating the particular process under study, right? Right. Like what are the important elements? What are the pieces that you can put together? And just having sat in, Glenprint, that's James Glazer's, seminar series, like that those are the kinds of things that they seem to focus on, like what are the important elements that I can simulate and get at some sort of mechanistic model, interpretable model, etc. But I'm enjoying the conversation, and and now just still starting the day here on the West Coast. So forgive me, but I'm having breakfast.
All right. All right, and then we had some people here working on Barshan and Jayadratha on graphs and developmental graphs. I don't know if they wanted to say anything, if they had any input.
Hello, am I audible?
Yeah.
Okay. So let me give a small intro about myself. I'm Barshan. I'm an final undergrad from Amrita, which is in India. So I heard this whole conversation, I had a bit of doubt. So as Bradley said, that there are a lot of these models which are trying to simulate the world, and all of them are developing it on different concepts, and some on biology, some on physics, some on chemistry. So under the same environment and the same conditions, so would they contradict each other's movement or what they are acting upon? So that is possible, right? That all of them are contradicting whatever the other one is. So maybe we could create something and find similarities in all of those models and create a better simulation out of that. I think that would be a great thing to go for. I mean, that's just an overview, idea I got.
Yeah.
Yeah, I mean, if there's something useful in some models, so models can be combined, right? Like if there's a lot of, I guess, code that's written for simulating some particular thing in a worm. Maybe we can just tell AI, get this module and integrate it into this common model, right? So there could be common model that brings together everything that's useful from all other models. And it's currently quite easy, like this AI program, because one human could be not able to do it because it's like a lot of work. But AI, we can just ask AI to do that, and it becomes much easier. So one can do like 10 times more than she could do before. So if there is some particular model that you want to suggest that you think is good, you could post it, and I could look into it if I can borrow something for my model, for example, from that model.
Yeah. What language is your model written in, or what do you usually work with in terms of?
It's C++.
Okay,
C++. Yeah. But if there's something on Python, I'm sure like it could be all converted. I use C++ because it's very high performant.
Right.
And it looks like the calculation can become a bottleneck very fast. So since I want to go as low as possible, I need to language that like provides the best performance.
Right. Yeah, I think C++ is a good choice for the kind of simulations. I just wondered, since you're working in NVIDIA, have you parallelized a lot of some of the stuff? Or are you planning?
Yeah, I didn't yet. So I'm so right now the simulation runs faster than real time, right? So I didn't have much incentive to like even use multiple cores. Right now everything is on just one CPU core.
Okay.
But I do envision when it gets more complicated, because right now I have like just few genes, few proteins flowing around, so it's not a big deal to simulate. But I guess I will start thinking about parallelizing. Yeah, soon.
Yeah. Well, that's just always a question. Yeah, no, this is good because we of course have limitations of computational power and trying to figure out how to build a model without you may have access to a lot of computational power, but it depends on like if you want to run it locally or if you want to run it as like an individual. People might not have that kind of power to run their own simulations. So it's always a thing you have to think about, kind of.
Yeah, right. So the compute it still costs money. So everything like if I need a lot of compute, then it will be expensive, and then we come to the problem of financing, how to finance this all, right? So to finance this, I need some application that can be useful to people. Like if I just show this very simple simulation, nobody can really use it for anything, right? It has to become useful. So I'm looking for some application where I can say, oh, if you do this, you can earn money with it, right? So, and for example, I'm looking at there are a lot of companies that use biological organisms to synthesize things, like for example food, right? People build artificial egg whites, for example, or some kind of biofuel, or for example protein for artificial meat, right? So if we can use this model to somehow improve the output of this production, like I can help these people to optimize their production and to reach higher volumes of production with the same money. Then I can get more money, and this will open new possibilities, right?
Yeah.
So if you have ideas how to use this, for example, I wanna. So right now my repository just has worm simulation. But I want to add yeast simulation because a lot of companies use yeast for like bioproduction.
Yeah.
And then I want to try to like simulate some metabolic model basically. So there are a lot of metabolic models. I want to add it to the repo so I can use it to pitch to these kind of companies.
Yeah, I think that's good.
I'd be very interested in the yeast and metabolic models.
Oh, really? Okay. Then I can ping you and like when I have some questions.
Yeah. I got some people here, so I'm in a synthetic biolab here in San Francisco. So those are the kinds of things that people would like to see.
Oh, nice. Then I can drive there because I'm in Los Gatos.
Oh, okay. Gotcha. Sure. Okay. Yeah, we're right downtown, San Francisco.
Okay, nice.
Yeah.
Right. Yeah, and then I just wanted to mention that, of course, there's a lot of this work being done on whole cell modeling, where people are simulating different types of bacterial cells, and the idea is to get as much detail about the cell as possible, the kinetics, and then simulate like a certain amount of the of time that goes by in the dynamics. So there are a lot of there are a couple of groups in the world that are working on this sort of model where they model every sort of component of the cell at sort of like the protein level, DNA level, and then have like one entire cell, and then they simulate the dynamics. And that, of course, takes a lot of computational power. So they're simulating like maybe a second or 2 seconds of dynamics. So that's kind of interesting stuff because it's like you get this really high level of detail, and then you can simulate it for a little while, and that's good enough to sort of understand what's going on. And then, of course, that contrasts with like simpler models, which maybe you can run for longer. Maybe you can run it for like the full course of like C. elegans development. But you couldn't, of course, run like a whole cell model for that long to simulate the divisions because it would just be way too much. You'd need way too much computational power. But of course, as you mentioned, we're kind of getting more and more computational power online where we can do this sort of thing. Yeah, so it's really interesting. The potential, that's really what we want to look at. Yeah. So do we have any other questions?
Yeah, is there still some time left?
Yeah, go ahead.
Yeah, actually, Shreyas built a system. He want to share that his work on how connectomes of C. elegans develop in time domain using graphs and neural ordinary differential equation. He created a generative model that can show some intermediate time steps of C. elegans, its neuronal connection development. So if Shreyas is ready, please go ahead.
Hi everyone. Just introduce myself. Hello. I'm Shreyas. I'm currently a third year undergrad at IIT Madras. It's a college in India. So regarding the presentation which Kirill gave, it was like quite interesting to see the visualization of the embryo. And regarding the point which we were discussing, like a one model for the whole thing, I kind of with Hussain, I agree that I guess the discrete model can be good, but should I proceed with the presentation?
Okay. So just I guess we're transitioning, and so thank you for presenting, Kirill. We had a lot of interesting, and we'll continue on if you'd like to attend future meetings. You're more than welcome, and we'll keep talking about your model and thinking about how to use it, maybe in different ways.
Okay, thank you.
All right, thank you. So Shreyas, would you like to present on your things?
Yeah, sure.
Okay.
Okay, so I'll start the presentation. So the data I've used is the Wheatwillet data in this GitHub repository. It's basically an Excel sheet where there is I'll just show the data. It's of this type. So there's pre, post, and a type and a synopsis, the number.
Yeah, so this is the data which we can see in this GitHub repo. So I would first like to discuss the article from which I used this data, what it says. So it says that it discloses many ground truth biological facts about the connectome and how it is structured, some of which can be relevant and are mentioned below. So it's a symmetrical wiring. So the increase in synapses during L1 is the basic Elegans with the baby embryo. So during that, the left and right wiring increases symmetrically. So this is something which can be useful for us. So the new connections are more likely to form between neurons that are shared larger physical contact areas at birth. So it sort of like forms a clusters around the larger physical contact areas, and so each connection is classified as stable, development dynamic, and variable. So like say if a connection is stable, that connection would go on for the entire connectome interpolation, across all the stages. If it's variable, the connections could change in between, right? So this is something which I thought can be useful and can be used as a biological bias. So yeah, these are the important facts which I found from the paper, and yeah, I'll proceed. So yeah, this is something which is interesting. So like from this figure, we can understand that there are various types of neurons which are connected in a particular hierarchy, right? So the hierarchy goes like that. Is this visible or should I like zoom away?
Yes, it's fine. Yeah, it's visible.
It's fine. Okay, cool. Yeah. So we can see that it goes like sensory neuron, then interneuron, and then motor, and then muscles. And here, like we have a hierarchy build up, and then these are the way the neurons are connected. So these are the four biological facts which like these are not like inclusive of all the facts. These are like which I found can be relevant. So which I will try to use in my model to like have a biological bias. Because just having mathematical bias, I don't think would work that well. Having a biological bias would be good, as we do in physics-informed neural networks. So yeah, so the data pre-processing. So the initial data is in the format shown on the right. This is the initial data which I had. And in total, 8 stages of C. elegans connectomes are given. The initial 4 stages are a part of the L1. The next 2 are L1, L2, L3. And the following next 2, like the 8th and the 7th and 8th file of data, are the adult. This was written in a paper. And then, so this data is converted into adjacency matrix. So like what is adjacency matrix? It's basically a n cross n. Say I have 225 neurons. So it's basically n cross n matrix, and in each cell is written if the neuron is connected to a particular neuron, and like what is the strength. Say it has 5 connections between those. So like basically adjacency matrix is created where all the unique neurons are the rows and columns, and the number of connections between each pair is written in the corresponding cell. So later the matrix is converted to binary for simplicity, because as of now, currently I'm just modeling if a connection is there between 2 neurons, and later I'm scaling it to the strength, like how many connections are there. But as of now, I'm just doing it binary, like if a connection is there or not. And then a mapping graph is created for GCN. So what is a mapping graph? I'll just show it here. Yeah, so like basically this is a mapping graph. So like say this is 0, 0, 1, 0, 15, 1. So like it basically signifies that 0 is connected to neuron 1. Like neuron 0 is connected to neuron 2, and so on. So yeah. So I'll just go through like what this is. This can be like uninterpretable as of now, but I'll just go through. So for showing, I showed like the mapping graph, which basically maps the since it is a graph, so we need edges and like message passing. So to understand which neuron is beside which one and what are the connections, that mapping graph is created. Okay, so I'll go to the methodology next. So the methodology is broadly divided into 2 parts, which is the GCN, the encoder, and the Neural ODE. Decoder is just a small part of it. So yeah, so what the GCN does is like it's a sparse graph connection. It basically tells who connects to whom, and the input is the adjacency matrix, which I previously told you. So here I've used the term identity adjacency matrix. So what does that mean? It's like all the neurons are stacked in the rows and columns, and it's an identity matrix. So all of 1, 1, 1 in the diagonals and rest are zeros. So why this is so? So basically what we're doing is like we are having a column for each neuron. Say this way, right? So this is each neuron. This is the adjacency matrix, the initial adjacency matrix. I just wrote in my iPad and put a screenshot here because it was difficult to like, type this out, everything. So yeah, so this is my input adjacency matrix, where diagonals are ones and the rest are zeros. So the values of this will change and evolve through the graph neural network, and it will learn its neighborhood. So this is the motto of it. Then the action. So feature sharing is done, as I told. So it will look across its neighbors, like who all are around me, and it will change its vector. We can say like each neuron has a vector, and it will change its values. And then it learns how to compress the graph topology, each column of adjacency matrix, into a meaningful starting coordinate z star for the neural ODE. Right. So now I'll go in the mathematical terms of the graph convolutional network. So yeah, this is my input to it. So again, the edge index, as I told you previously, this is my map as to I guess this is like I basically wrote 2 here. It should be 15, like 0 connected to 15, neuron 1 connected to 4, and so on. This I by mistake wrote 2. My bad. So this is basically why we're having 1, 2, 3 in a diagonal fashion is to identify each of the neuron in a unique way. It's like basically identity of each neuron and the ID of each neuron. And then these are my inputs. The tensor, this is created from the data which I have, the Excel sheet which I showed previously. And then the left side is my adjacency matrix. Now each column of the adjacency matrix is processed individually and updated. So these are each processed individually. So as we can see, this is the initial. Say this is A0. Now this is my A1. So A1 is basically this function really of feature, like this feature plus its neighborhood sum into the weights. So W is what is learned. So this is my graph. So say for 0th is connected to the first one and to the 15th one, and say something else later. So this will basically model, okay, I'm connected to so and so. I have a weight which I have to learn, and it will learn it. So this learning will be done along with the neural ODE. So this is like there is not any individual loss function on the GCN. GCN plus the neural ODE altogether has a loss function in the end. So I'll come to it later. So my output is basically 16 cross 225 compressed ridge matrix. So my vector length decreases from 225 to 16, and the feature vector of each neuron has learned the info about its connection and its neighborhood. I'll just stop here for a second if there's any doubt in the graph convolutional network, because again, on this is what neural ODE is built on.
That looks good, yeah.
Right. So yeah, I'll come to neural ODE now. So we assume that the connectome, just to come back, like this output of this is the input for the neural ODE. So we assume that the connectome latent space, which is the output of GCN, I'll call it connectome latent space, since we have compressed it, is the latent space, is governed by a continuous time dynamical system. So this is the assumption, which can be a flaw of it also, but yeah, as of now I'll just assume it. And then we define an initial value problem integration, like everyone knows initial value problem. So this is basically like I'm modeling how ZT. Okay, so I guess I did a abuse of notation. I didn't write here ZT, but yeah, this matrix which is compressed and 16 cross 25 size, that is the ZT. That is my ZT. Okay, so it evolves with time. So d of ZT by dt is, I'm assuming it's a function of ZT and time, and theta are my learnables. So as you can see, t here are the continuous variables, time continuous variables, and f theta is a learnable vector field parameterized by the weights theta. So it's basically a neural network. As you can see, f theta is parameterized as a multi-layer perceptron that operates on node feature. So what is node feature? Node feature are basically these features. So these learned features, it will act on those individually. So yeah, these are my equations, which are the initial values. This is my initial value for solving this. So I'll go through step by step. So the first step is temporal concatenation. So since time is the important factor here, so I'll concatenate temporalness in each of the vectors. So Znt is, so if this is my Zt, this is my Z0t, Z1t, Z2t, and so on. So yeah, I guess I should have wrote notations in a bit clearer way, but yeah, my bad. So yeah, again, so this is my concatenation. So it's a dimensional plus 1. We are adding a dimension to it. So once this is done, I do the transformation layer of MLP. So this is basically tannage activation function. I'm going through 2 tannage activation functions and like basically 3 layers. But I just did 3 layers because I had some limited compute, but I can scale it if I have like time and some compute. I'll scale this. Then numerical integration. So since my output here, as you can see, is dzn by dt. So I'm training for that. Like that is my final output from the MLP. So I just integrated using my initial value to get ZTK, which is like the connectome at any point of time, right? So now this ZT won't be directly the connectome, it will be the latent space. I'll show how the decoder like builds it into the connectome, back to the connectome. So you can see this seems a bit cryptic here, the F theta, Z tau, comma tau. So this essentially, if we look here, is just the as you can see, this is this. So this is just the essentially is the dzn by d tau. So if we integrate it, we'll get dzn, and then we add to this, and essentially then we get solving the initial value problem. We get the latent space of connectome at any point t in time. So using this, we can yield z at any time step. So decoding is basically in a product. So if you take ZT inner product with another ZT, like say it's a element-wise sigmoid, in an element-wise sigmoid. So like I have 225 ZTs, and then I'll take inner product of each one with each one. So like basically I'll have 225 C2. C2 as in I'm selecting 2, that many inner products I'll have, and then I'll see how the each of the vectors are like correlated with each other. And then that passing it through the sigmoid, which gives me a probability matrix of n cross n. So now what my output is, it's a probability matrix n cross n, where each of the value is the probability if there's a connection between those neurons or not. So that is what is my output, right? So coming to the loss function, So coming to the loss function, so I'm using a bind. Okay, before I go to loss function, is there any questions on ODE, the neural ODE part?
No.
Okay, cool. Coming to the loss function. So I'm using binary cross entropy logic loss because my outputs as of now are zeros or one as to like if connection is there or not. So that's why I'm using a binary cross entropy loss. And then so the interim time steps. So as I said earlier, like I have L1, L2, L3, L4, and so on. So those interim time steps are considered as labels. So my input to the model is just the L1. To the graph model, and then just that is the input. Others are not shown to the model. It's just used as labels, and then like interim points. And so this is my loss. This is not quite good. I mean, it's just like it's a mid, but fine. I wish to improve the architecture and better my things. So I'll tell you how I'm wishing to do it. So in the heat map visualization, which I told, but this doesn't tell us much. We need something better. So this is my graph visualization which I've done. So only the connections with probability 0.8 is kept. If I keep all of the connections with like say 0.5 plus probability, the graph gets very messy and difficult to interpret. So I just kept connections with probability 0.8 plus, which gives us some guarantee. So the colors are not explicitly programmed with biological names as such like sensory neurons or motor neurons, which I told in the biological paper which I referred in the start, as to like they told that each of the neurons are categorized into some spaces with their sensory or motor and so on, and there's a hierarchy. But so the colors are not programmed to be that. They're determined by like community detection, specifically the greedy modularity algorithm. But the colors visualize functional modules. Like though we didn't tell it, but it understands that okay, here is a cluster, see the yellow ones, the orange ones, here is a cluster where these neurons are a lot connected amongst themselves and they form some cluster in itself. Like there are not many connections from say orange to red or so on. They form a cluster in themselves. They're remaining with each other. Like as I told, we don't tell the model like it is a motor neuron as such. Like for example, I'm not exactly pointing out a motor neuron as such. I'm just giving an example. Then the math is realizing that okay, hey, these 20 neurons are a lot together. Like they hang out together. They just are connected amongst themselves. So they can be certain type of neurons. So it's figuring out clusters in the connectome. And I just show zoom. This is a larger version of this, the graph. That's the zoomed version. So in zoomed version, each of the neuron has a name. As you can see, this one, the point which I'm pointing is like ABL, and its degree is 48. Like it's connected to, there are 48 connections to it. Like from some other neuron to that. There are 48 connections. So I have created the graph for all the interpolations. So what I've done is t equal to 0, t equal to 0.5, 1, 1.5, and so on till t equal to 3. That interpolation I've done. And yeah, any doubts as of now, or I'll go towards flaws and future works.
That's good. Yeah, why don't we do the flaws and future works?
Yeah.
I think that looks good.
So for the flaws, what I'm thinking is I'm not taking, as I stated earlier, I should take into account biology. What I'm not taking into account as of now is the biology part. Like I have just used math as of now. So I intend to use hyperbolic neural ODEs. So why hyperbolic neural ODEs? The paper describes a clear hierarchy as to sensory to interneuron to motor and so on. So the standard Euclidean space, the normal vectors, is bad at representing hierarchies, trees. So the so we can map the neurons into hyperbolic space, say Poincare Ball, and instead of Euclidean space, and the hyperbolic geometry, it naturally represents trees and a hierarchy. So if we compute in that space, maybe we can have a better representation of hierarchy and a better outcome. Second thing is BC laws only cares if there's if an edge exists or not in the brain, and it doesn't care for a functional circuit, like if the circuit is actually functional, if it actually makes sense. So we can add a regularization term to the laws that penalizes graph properties. Like what I mean here is that this can force the model to match the degree distribution, like preventing it from creating random edges and forcing it to create the hubs like AVA, APB. So what I've seen in papers. So what I mean here is like paper states that there are hubs of neurons. It's not like, as I stated, like there are like physical, if there is a physical connection, it intends like a neuron is more probable to connect there if there is a more physical contact connection, and it creates hubs. So there are like multiple hubs in the connectome. This is what was observed from the paper. So I intend to create a regularization for that part as well. So again, the flaws of the differential equation which I use is too basic, as I stated. Like it's just dz by dt equal to some MLP, but there can be like degree 2 connection as well. So I intend to like use a better differential equation. Third thing is the GCN assumes every neighbor is equally important, whereas the paper states that some connections are stable, always present, whereas some connections are stochastic, variable, which is stochastic. So I need to get a way around. So this is in this part I didn't write here. I was thinking I can use a graph attention layer. I can add in the graph somewhere to pay attention towards which neighbors are actually important and which are not that important. This is the third thing, and the fourth thing is I need to find more data set to verify this idea, and maybe if possible, like I don't know, like maybe get a new organism to test this as well, like if this is a generic thing or this is limited to C. elegans. So basically I need to find more data sets. I did find some more data sets but it's of some other organism, not of this particularly. And like the future work, I need to inject the anatomy into this, like using this data set to tell the model which neurons are sensory versus which are motor. As of now, I'm not telling the model which are sensory and motor. I'm trying to interpret it through the model, but I can also tell the model like these are sensory and these are motor. So like my interpolation can be a bit more better. So I also intend to add geometry like penalizing long connection in a loss function. So like, again, from the paper, it was interpreted that long connections are not that it doesn't occur that often. So we can penalize long connections. And the clustering part which I said, I forgot to add it here, the clustering part as well. So you can analyze dynamics. So using the model to find the exact critical periods of wiring changes that are missed between static snapshots. So currently the connectome is like we have snapshot, we don't have interpolation. That was the whole problem statement. So what we can take from this solution, like what I can try to do is that at what points of the life of the C. elegans, what are the critical periods in which connections are changing, when is the brain developing suddenly and all. So that can be done. So what I'm trying to do is as I said, I have all the graphs of each of the time step. Like I can generate graph of any time step. So I'm trying to create an animation now as to show how the brain is evolving, and then we can observe at what point exactly that's happening. I have generated animation, but it's not that good. I'm just trying to get a good animation out of all that. So yeah, and then finding and testing on new data sets, or maybe find a new organism. So yeah, that's all from my side. These are the references. This is the paper which I used, the C. elegans Connectome paper. This is the GCN paper. This is the Neural ODE. So basically, I tried to combine these two papers and create something new. This is not entirely new, but I'm just combining two papers. And then this is the implementation code. I'll share it in Slack, and you can like visit the code and play around with it maybe. So yeah, this is from my side. Any questions?
Okay. Yeah, thank you. That was good. Yeah, I'm looking forward to seeing the visualization. I think you have a lot of really interesting methods there. Definitely the neural ODEs. This is just kind of looking at the evolution of a connectome and kind of getting a sense of what that looks like, the dynamics and that. And yeah, looks like you've got some really good work already, and visualizing it, I guess, will be key. I don't know if Morgan had something to say.
No, I've been playing with Neural ODEs recently, but yeah, this is interesting, and I'll try and play around with this.
Yeah. So what are you using? Oh, go ahead. So what are you using for your neural ODEs? What kind of software? Is it just in Python, or is it a toolbox that you're using?
Sorry, is that for me?
It's for Shreyas. Well, for you too, I guess.
Yeah.
But
That's not
Go ahead.
Okay, so I'm using a library in the Neural ODE part. It's a part of I guess it's a part of Python geometry, the Neural ODEs, and there I'm using it in Torch only. Like I'm using it in Torch.
all right.
But there are some mathematical softwares like I guess Sage and all which do it, but I'm not quite familiar with it. I was familiar with PyTorch, so I did use it.
Yeah, it's best to stick with PyTorch, I guess.
Yeah, I'm more comfortable with PyTorch, so I stuck with it. But there are some mathematical libraries as well.
Yeah, I'm doing everything with Jack's libraries.
All right. Yeah.
Yeah,
that's good. All right. Yeah. Thank you, Shreyas. That was very impressive. Yeah. So why don't we call it a meeting? Thank you again to Kirill for presenting. If you look in the chat, he's left his email address if you're interested in getting in touch with him. And we'll probably want to do some sort of collaboration. And so people, think about what you would like to model. The open source repository doesn't have a link to it here, but it's not public yet. So we'll definitely get in touch on that. And then Shreyas, thank you for your presentation. Hopefully, I guess we might want to follow up next week on it as well.
Yeah, sure.
Yeah.
I'll get the animation up. Yeah.
Okay. And then I think Barshan probably wants to present, but we'll do that next week. We'll just keep presenting stuff.
We can do that next week. Yeah, we can push it. Yeah.
Yeah.
And then get some more results now.
Yeah.
Okay.
All right. Thank you. Have a good week.
Okay, I'm interested in modeling too, especially using some tensegrities
Okay.
in with the model. So.
All right. Yeah. All right. Thank you.
Okay.
Yeah. Thanks everyone. It's great to meet you after a long time. Thanks.
Oh yeah. Nice seeing you again. Yeah.
Yeah.
Thanks.
Bye.